This is SOSF. Man, my brain is still buzzing from the stuff I read this morning. We spend so much time thinking about external threats, right? Like, what's in our food, what's in the air, all the crazy stuff happening out in the world. But what if I told you that the most complex, high-stakes war imaginable is happening inside your own body, 24/7? And you are the battlefield. I'm not talking about just fighting off a cold. I'm talking about a full-blown civil war that's constantly on the brink of erupting, and the only thing stopping it is a secret police force you never even knew you had. This is not science fiction, man. This is the reality of your immune system, and a few brilliant minds just got the Nobel Prize for finally figuring out how these gatekeepers of the self actually work. It's a story that is going to completely change how you think about getting sick, about cancer, about everything. So, let's break it down. Your immune system. Think of it like the most hardcore, elite military force on the planet. Its job is to seek and destroy. It's got soldiers, these cells called T cells, that are just stone-cold killers. They float around your blood looking for anything that isn't you. A virus, a bacteria, a mutated cell that could become cancer. If it's an invader, a non-self entity, the T cells are designed to go absolutely medieval on it. Annihilate it. No questions asked. And thank God they do, right? Without them, a paper cut could kill you. We'd be walking petri dishes. But here's the billion-dollar question. The one that stumped scientists for decades. If you have this army of ruthless killers programmed to destroy anything that isn't you, how in the hell do they know not to kill you? Your heart is you. Your liver is you. Your skin is you. Why aren't these killer T cells just constantly waging war on your own organs? Because if they did, you'd have what's called an autoimmune disease, where your body literally tears itself apart. It's a real and devastating problem for millions of people. For the longest time, the best explanation scientists had was this idea of a boot camp. They called it central tolerance. Inside your chest, you have this little gland called the thymus. It's like the Paris Island for T cells. When T cells are just little cadets, they go to the thymus to get trained. And the main part of their training is this brutal test. They get shown pieces of your own body, your own proteins, your own self. And the rule is simple. If a T cell cadet shows it's a little too interested, a little too aggressive towards your own stuff, if it binds too strongly to self, it gets executed on the spot. Programmed cell death. They basically identify the psychos, the potential traitors, and eliminate them before they can ever get out into the field. These were called the forbidden clones. It's a hardcore system. The T cells that graduate are the ones that are good at killing foreigners but will leave their own people alone. And for a while, everyone was like, okay, that makes sense. Problem solved. But there was a huge, gaping hole in this theory. The boot camp isn't perfect. It's a leaky firewall. Think about it. The thymus can't possibly show these cadets every single part of your body. What about proteins that are only found in your brain? Or your pancreas? Some self-reactive T cells, the ones with a lower-grade craziness, they slip through the cracks. They graduate from the academy and go out into your bloodstream, fully armed and dangerous, with the potential to start a war against your own tissues. So, if these potential traitors are always circulating in our bodies, and they are, why aren't we all just dissolving into puddles of autoimmune goo? Why is a healthy immune system the norm and not the exception? It was a massive paradox. This is where the story gets really wild, and it involves three incredible scientists from totally different worlds who were chasing this mystery from opposite ends. You've got this Japanese immunologist, Dr. Shimon Sakaguchi. This guy was a real maverick, man. Back in the 80s and 90s, the idea of a suppressor T cell, a cell whose job was to calm things down, was basically a joke in the scientific community. People had tried to find it before and failed, so the whole field was considered a dead end. But Sakaguchi was obsessed with this paradox. He just knew there had to be something else going on, another layer of control. So he does these incredibly elegant experiments. He takes newborn mice, and he removes their thymus gland. He noticed something crazy. If he did the surgery on day 1 of life, the mice were just immunodeficient, which is what you'd expect. But if he waited and did the exact same surgery just 2 days later, on day 3, the mice grew up and developed a whole spectrum of horrible autoimmune diseases. Their bodies just went to war with themselves. It was a brilliant deduction. Sakaguchi realized that between day 1 and day 3, a special kind of cell must be graduating from the thymus and going out into the body. A cell whose whole job is to keep the peace. A protective cell. In the absence of this cell, the other killer T cells that had already populated the body just went haywire. To prove it, he did the next logical step. He took T cells from a healthy adult mouse and injected them into one of the mice that was destined for this autoimmune disaster. And boom! Nothing happened. The disease was completely prevented. He had proven, definitively, that there exists a population of cells, these peacekeepers that are actively, constantly patrolling your body and stopping your immune system from attacking itself. He published this in 1995, basically reviving a dead field of research and laying the groundwork for everything to come. He found the WHO. Meanwhile, across the Pacific, near Seattle, two American scientists working in the biotech industry, Dr. Mary Brunko and Dr. Fred Ramsdell, were tackling the exact same problem from a completely different angle. They weren't looking at cell functions. They were gene hunters. They were studying this specific strain of mutant mice called scurfy mice. And these poor little mice were a train wreck. They were born with an immune system that was stuck in overdrive. It was just expanding like crazy, attacking their skin. That's why they were called scurfy, attacking all their organs, and they died within a few weeks. It was a catastrophic, real-world example of what happens when the immune system has no off switch. Brunko and Ramsdell knew this had to be caused by a single, broken gene. So they went on this painstaking genetic safari. This is tedious, meticulous work, man. They mapped the mouse's X chromosome and narrowed it down, and down, and down, until they finally cornered their suspect. In 2001, they published a landmark paper. They found it, a tiny 2-base pair mutation in a gene that nobody had ever characterized before. This tiny error was causing the entire immune system to have a fatal meltdown. They named the protein this gene makes scurfine, and the gene itself is now known by a much more famous name, FOXP3. And here's where it clicks into the real world. They realized that a rare and devastating disease in human baby boys, called IPEX syndrome, looked exactly like what was happening to the scurfy mice. And sure enough, they found that IPEX is caused by mutations in the human version of that exact same gene, FOXP3. They had found the master switch, the one gene that holds the entire system of self-tolerance in its hands. You break that one gene, and the whole treaty between your immune system and your body is off. They had found the what, so now you have these two massive puzzle pieces. In Japan, Sakaguchi has identified a functional peacekeeper cell, but he doesn't have a unique way to identify it, no molecular name tag. In America, Brunko and Ramsdell have found the master gene for immune peace, but they don't know exactly which cell it's controlling. The grand synthesis, the big aha, a moment, came just a couple of years later. Sakaguchi and other labs, now armed with the knowledge of FOX3, put 2 and 2 together. They tested Sakaguchi's peacekeeper cells, and wouldn't you know it, they were packed to the gills with the FOX3 protein. It was the name tag. It turned out that FOX3 isn't just in these cells, it's the master regulator that builds them. It's the transcription factor, the foreman, that tells a T cell, forget being a killer. Your new job is to be a cop, a peacekeeper. These cells were officially named regulatory T cells, or TREGs for short, and the whole picture snaps into focus. The tragedy of the scurfy mice and the boys with IPEX syndrome was that a genetic typo meant their bodies couldn't manufacture this internal police force. Without the TREGs on patrol, the slightly leaky boot camp in the thymus becomes a fatal flaw. The few self-reactive killer cells that slip out have no one to stop them, and they start a riot that burns the whole city down. This discovery was a complete earthquake in immunology. It changed everything. Our understanding of health was transformed. Being healthy isn't a passive state. It's not just the absence of disease. It's an active, fiercely maintained truce. A dynamic equilibrium that is constantly being policed by these TREG cells, all orchestrated by the master switch, FOX3. This is peripheral tolerance, the policing that happens out in the streets of your body, not just at the central boot camp. And it's for this monumental discovery, the convergence of these two lines of research, that Sakaguchi, Brunko, and Ramsdell won the Nobel Prize. Now, this is where it gets really applicable to you and me. Because once you identify the police force, once you understand who's in charge of keeping the peace, you can start to manipulate it. And this has opened up a therapeutic strategy that is a total paradox, a real double-edged sword. Because in some diseases, these TREG peacekeepers are the heroes. But in others, they are the enemy. Let's talk about cancer first. A tumor is one of the smartest, most diabolical things in nature. It's not just a dumb lump of growing cells. It learns. It evolves. And one of the most brilliant things it learns to do is exploit this very system of peripheral tolerance. A tumor is, technically, self. It grew from your own cells. But they're mutated, dangerous cells that the killer T cells should recognize and destroy. So what does the tumor do? It basically hijacks your body's 911 system. It sends out chemical signals that are like a siren call to TREG cells. It essentially screams, "Help! Police! There are aggressive killer T cells here trying to hurt me. Come protect me." And the TREGs, just doing their job, flood into the tumor. Once they're inside, they create what's called an immunosuppressive microenvironment. They get in there and tell the killer T cells, "Stand down, boys. This is a protected site. Everybody calm down." They essentially form a protective shield of peacekeepers around the tumor, protecting it from your own immune system. It's like a crime boss who has the local police department on his payroll. And doctors found that the more TREGs you find inside a person's tumor, generally, the worse their prognosis is. So, the goal of modern cancer immunotherapy became clear. You have to break this shield. You have to somehow get rid of the corrupt cops inside the tumor so the real soldiers can go in and do their job. This is the basis for some of the most successful cancer drugs on the market, the checkpoint inhibitors. One of the first big ones, a drug that targets something called CTLA-4, is now understood to work in large part because it preferentially kills TREG cells inside the tumor. Because TREGs have a ton of this CTLA-4 on their surface, the drug is like a guided missile that takes them out. You remove the peacekeepers, you remove the brakes, and suddenly the killer T cells are unleashed and can attack the cancer. And the proof that this is how it works is, ironically, in the side effects. What happens when you give someone a powerful drug that systemically weakens their TREG police force? Their immune system starts to get a little trigger-happy. People on these drugs often develop immune-related adverse events, basically, drug-induced autoimmune diseases. Their immune system starts attacking their skin, their colon, their glands. It's a clinical validation, in humans, of the Scurfy mouse experiment. When you lose the peacekeepers, chaos follows. It's a fine line to walk, but it has revolutionized cancer treatment. Now, let's flip the coin completely. What about actual autoimmune diseases? What about type 1 diabetes, where your immune system is killing the insulin-producing cells in your pancreas? Or rheumatoid arthritis, where it's attacking your joints? Or lupus? In all these cases, the problem isn't too much peace, it's too much war. Your TREGs are either outnumbered, or they're not functioning properly. Your internal police force is MIA. So the therapeutic goal is the exact opposite of cancer. You don't want to kill TREGs, you want to reinforce them. You want to call in the National Guard of Peacekeepers. This has led to one of the most futuristic ideas in medicine, adoptive TREG cell therapy. It's basically creating a living drug. The process is insane. They take a sample of your blood, and they use these incredibly sophisticated machines to sort through millions of cells and pick out your tiny population of natural TREG cells. Remember, they're pretty rare. Then, they take this small handful of your own peacekeepers and they grow them in a lab. They give them all the nutrients and growth factors they need, and they coax them to multiply, and multiply, and multiply, until they have billions of them. A whole army of your own, custom-grown peacekeeper cells. And then, they take this massive population of TREGs and infuse them back into your body. The idea is to fundamentally shift the balance of your immune system back towards peace, to send in enough cops to finally quell the riot that's been destroying your tissues. This isn't a theory anymore. This is happening in clinical trials right now for type 1 diabetes, for lupus, for preventing the rejection of transplanted organs. Imagine getting a new kidney, and instead of taking a lifetime of harsh, system-wide immunosuppressant drugs, you get an infusion of your own TREGs that are trained to specifically tell your immune system, hey, that new kidney is a friend. Leave it alone. It could be a complete game changer. It's moving from a sledgehammer approach to a scalpel. Of course, it's not that simple. Nothing this complex ever is. And this is where the field is heading now, into the world of bioengineering, trying to solve the big challenges. The first one is specificity. It's a double-edged sword. With cancer, you don't want to kill all the TREGs in the body. You want to kill them just in the tumor. With autoimmunity, you don't want to cause system-wide immunosuppression that makes you vulnerable to infections or even cancer. You want to boost the peacekeeper response only at the site of the attack. So how do you do that? How do you make these cells smarter? This is where they're borrowing tech from another hot area of cancer therapy, CAR T cells. They're creating CAR TREGs. A CAR, or chimeric antigen receptor, is basically a synthetic GPS and targeting system that you can engineer onto the surface of a cell. So you can design a CAR TREG that is specifically programmed to recognize a protein that's only found on, say, the pancreas cells that are under attack in diabetes. Now, you infuse these engineered TREGs, and they don't just float around randomly. They home in directly on the war zone, delivering their peacemaking payload exactly where it's needed most. It's like giving your cops a specific address and an arrest warrant. The other huge challenge is stability. It turns out the TREG identity isn't locked in stone. They have a degree of plasticity. If you take a well-behaved TREG and you put it into a really nasty, inflamed environment, which is exactly the environment of an autoimmune disease, there's a risk it can freak out. It can get overwhelmed, shut down its FOX3 master switch, and, in a horrifying twist, it can actually flip sides. It can shed its police uniform and become a pro-inflammatory killer cell, throwing fuel on the very fire it was sent to put out. That would be a catastrophic failure. So scientists are now using tools like CRISPR gene editing to try and lock in the FOX3 gene, to make the TREGs more stable and resistant to converting to the dark side, even in the most hostile environments. We are literally at the point where we are not just using cells as drugs, but engineering them to be better, more reliable drugs. The whole thing is just mind-blowing when you step back and look at it. The journey from a skeptical scientist in Japan studying mice, and 2 gene hunters in Seattle studying a weird mouse mutant, has led us to a place where we have a unifying theory for cancer, autoimmunity, and transplant rejection. They are all, at their core, diseases of a broken peace treaty. Too much tolerance in one case, too little in another. And by discovering the cellular and molecular basis of that treaty, the TREG and its FOX3 switch, these Nobel laureates gave medicine the keys. The keys to dial the immune system up, or dial it down. We've moved from a static view of our bodies to understanding that we are a dynamic, walking, talking battlefield, and that our health depends on an active, vigilant police force. The legacy of this discovery isn't just in the textbooks it rewrote. It's in the cancer patients who are alive today because of immunotherapy, and it's in the future hope for millions suffering from autoimmune disease. They found the gatekeepers of the self, and in doing so, they gave us the tools to finally start learning how to pick the locks. It just leaves you wondering though. A huge part of what we call aging isn't just getting wrinkles, it's the slow, steady breakdown of this internal policing. It's chronic inflammation, a weaker response to threats, and a higher risk of both cancer and autoimmunity. We're basically talking about an old, tired police force. So here's the thought I'll leave you with. If we master this technology and can give ourselves a perfectly balanced, eternally vigilant immune system, a police force that never gets old, never gets corrupt, and never gets tired, have we effectively solved aging? Or are we just clearing the deck for the next, deeper level of biological failure to emerge? Some bizarre cellular hardware problem that's currently so rare we don't even have a name for it. What does the end look like when your body's software is eternally perfect, but the machine itself is still designed to fall apart? See you next time.